Hello everyone, welcome back to another presentation. This is my presentation on GABA or gamma aminobutyric acid and how it works as a neurotransmitter and helps to provide anti-seizure medications. So we'll do a little background on GABA. It's a naturally occurring amino acid that works as a neurotransmitter in the brain, and neurotransmitters are what works as chemical messengers, so they can kind of relay information throughout the brain. GABA is an inhibitory neurotransmitter because it blocks or inhibits certain brain signals and decreases nervous system activity. By binding to the receptors GABA A and GABA B on neurons, it moderates and blocks impulses between the nerve cells, which reduces a neuron's tendency to produce an action potential, and this makes them less likely to excite nearby neurons. GABA attaches to a GABA receptor, which is a protein in the brain, and produces a calming effect. So this is what can help with feeling anxious, stressed, or fearful, and it can also help prevent seizures. It's become a popular supplement because not many food sources provide it in a natural way, but those that do are fermented foods, so like kimchi, miso, and tempeh. So what they've done is create a medication so that people can use this as a supplement in order to help relieve stress, anxiety, sensitive fear, but mainly the effects of seizures. So when misfiring occurs in GABA signaling, it is associated with certain neurological and psychiatric conditions. So when an action potential arrives at the axon terminal of a GABA-minergic neuron, it causes the release of GABA into the synaptic cleft, and then the following occurs. GABA binds to the target cell's postsynaptic receptors. The GABA receptors then open chloride ion channels in response to this, and the resulting rush of negatively charged chloride ions into the postsynaptic cleft or the postsynaptic neuron or the receiving neuron makes it more negative on the inside of the cell membrane, and this makes it less likely to fire on an action potential. GABA activates receptors very quickly within milliseconds, but the concentration of it then declines within the same time, within a few milliseconds as well. But the effect of the gabapentin is what lasts longer because the GABA unbinds from its receptor relatively slowly. So vigabatrin is the medication that is used to help alleviate the symptoms of seizures and alleviate what is causing seizures in the brain from the neurons misfiring. So in the brain, like I said, GABA has a calming effect, and in the spinal cord, this process allows for sensory information integration, which helps create smooth movements. So like when there's movements with muscles that can be kind of erratic, because this also has an effect on Parkinson's disease, but this helps to kind of calm those neurons down so that way the movements can become more smoother. The medication, vigabatrin, is used to inhibit the breakdown of GABA, which is effective in helping treat certain types of epilepsy. Tuberous sclerosis complex, or TSC, is one of the most common genetic causes of epilepsy. These seizures are generally present in childhood and are often associated with other neurological problems as well, like developmental delay and autism. And the majority of TSC patients have medically intractable epilepsy, but those who experience seizures in TSC are highly responsive to vigabatrin and have a 95% efficacy in stopping infantile spasms. So vigabatrin is like one of the only medications that has shown in recent studies to be effective against inhibiting seizures. The studies that they've done aren't entirely clear on why this is, but they're trying to look into it more in order to understand it, so that way it can be used as a medication to treat seizures in a better way. So this medication may represent a rare example of an education that has an efficacy for a particular disease or type of epilepsy. It has anti-seizure effects by elevating brain GABA levels via inhibition of its breakdown by GABA transaminase. And seizures they cause an acute inactivation of the mTOR pathway. And although vigabatrin is effective for seizures in TSC patients, the mechanism of this unique relationship, like I said, is poorly understood. It appears to have unique therapeutic properties in TSC for a few reasons that I previously mentioned, and it has been reported to improve the long-term outcome of neurological development in epilepsy in TSC patients. So the mechanism of vigabatrin is well characterized in animal models and cell cultures, mainly in mice. And as you can see, the Y-aminobutyric acid GABA synapses comprise about 30% of all synapses within the central nervous system, which underlies the primary mode of synaptic inhibition. So, vigabatrin was rationally designed to have a specific effect on the brain chemistry by inhibiting the GABA degrading enzyme, GABA transaminase, which results in a widespread increase in GABA concentration in the brain. This increase of GABA functions as a brake on the excitatory processes that can initiate seizure activity. So what vigabatrin is doing is basically stopping these the GABA from releasing too much in the brain, because when it does that, it initiates the seizure activity. So it blocks those in order to stop that, so that the brain can kind of control those excitatory pathways, and that way the seizures are lessened or stopped altogether. And vigabatrin was also designed to have a precise effect on brain chemistry by being enzyme activated, irreversible, and a selective suicide inhibitor of GABA T, which leads to an increase of GABA available in the synaptic cleft, which results in an enhanced GABAergic transmission. On this slide, I have just some structures of GABA, what it looks like, and then structure of vigabatrin, which you can see are kind of similar in their structures. And then this area here, let me just make it a little bit bigger. This is like the synaptic cleft that shows how GABA is reacting with the neurotransmitters here. So we have the receptor over here for GABA, and then we have the GABA neurotransmitters, which are just these little things. And this is the nerve cell, so you can see how they're kind of integrating themselves into the nerve cell, and that's how they are activated within the brain as a neurotransmitter. And then over here, we are using the Chimera program just to see the structure of GABA. And so you can kind of move it around and kind of see, you can zoom in, look at it. I always like to hide the ribbons here, so that way you can see, get a better view of it this way, because the ribbons kind of get a little jumbled up there, especially with this one. But this way you can just kind of see how it looks when it's all together, just to give you an idea of what the structure looks like. So I hope you liked my presentation on GABA and vigabatrin. And then down here, I just have the articles that I used in order to get this information. Thank you.